information, genome context, clustered microRNA, database links, and so on. So now in this other page below, we see mature sequence of HSA-Me29A5P. Are we there? Are we there? Just scroll down. You will see something mature sequence HSA-Me29A5P. Are we there? Go down. Have you seen? Yeah. Voila.
As you are doing, you're following it from the board, then you can see it. Yes.
Are we there? This is where we are. It's 5P.
When you look at the
it's no mistake. Are we there? Yes. There's 5P. We are with 5P. You people should follow what I'm showing on the screen. There's 5P. Okay? So that is its own accession number. And the previous ID, it used to be HSA-Me29A star. Okay? Which is different from the stem lobe. You see the previous ID of the stem lobe, it used to be HSA-Me29A. Okay? And we have said it's the premature microRNA. So that stem lobe will go down and then give rise to these two mature microRNAs that we have below. Okay? The first one is the Me29A5P. Its previous ID used to be HSA-Me29A star. Okay? This star, in the past, this nomenclature used to tell you that this microRNA is the one that is expressed in a lower amount in the system. When you see this star, okay, it used to tell you that this is the microRNA that is expressed in lower levels in the system. And then they have given you the sequence. Okay, that is the sequence. So, if we want this sequence, we can click on this get sequence. That is why we came to this website also. We click on get sequence. Are we there? Click on get sequence. So this is the sequence that they have given us. So we can copy. This sequence is the sequence of HSA-Me29A5P. Okay? Which used to be 29A star. Okay, so this is the current nomenclature. So we click and we go back. I just wanted to show you the sequence. So this is the sequence up here. If you want to copy, you just click on get sequence and you go and you copy on that page that will open up. Are we there? Okay. So evidence, experimental, cloned, and all of that and all of that. So predicted target, minor, that is it. Target, scan vet, that is it, and all of that. So what is interesting us here is that this name they have given us above here is the current nomenclature. The previous nomenclature used to be this. And I've told you that this tag will tell you that out of the two mature microRNA, this one is the one that is least expressed in the system. And it might likely be cleaved away, or it might remain in the system and have some target, okay? But the main one that will be functional will be this one below. Mature sequence of HSA-Me29A3P. Okay? You see the previous ID, they will give you as HSA-Me29A without an asterisk. Okay? Without an asterisk. So this is telling you that this is the one that is more expressed in the system. Are we together? But with this, our current nomenclature, we cannot distinguish which one is highly expressed and which one is in a lower concentration or is less expressed in the system. Are we together? With this, our current nomenclature, okay, you cannot tell which one is more in solution or is more in the system is more abundant in the system than the other. But with the previous nomenclature, we could easily predict which one was more dominant in or more abundant in the system. Okay, voila. So this is its own sequence. If we want to get the sequence, we click on Get Sequence. It is searching. The data started again. Anyway, let's continue. It is still searching. It will take us time, so let's just continue. Okay, so what I want you to know is that this is a sequence of, you can count it and see the number of nucleotides that we have. We said mature microRNA should have their RNAs with about 22 or 18 to 22 nucleotides, not so? So if we count this, it should be around that range. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Okay? So one thing we should also note is that this microRNA generally comes from the introns, the non-coding areas of the RNA. I mean of the gene. Sorry, of the genome. Okay? Genome or gene. They come from the introns. So in the past, they did not know about these microRNAs. They did not know about the function of microRNAs. They used to say that all the introns, they link up and they used to combine all the introns, and then they call them, if you do not code for a gene, they say you are junk. Okay? But now further research said no, these introns are not completely useless. Although they are usually kept away, at times there are some of them that code for these microRNAs. And these microRNAs, they function in gene regulation, okay, by regulating the expression of messenger RNAs. Okay? So, if you remember yesterday, I was talking about the coding region of the DNA. That the coding region does not just code for proteins. So those areas that also code for functional microRNAs, they also say they are coding. Okay? But in the past, they will say that the area of the DNA that codes for proteins is the only part that codes, the only part that was regarded as coding. But today, you also have those regions. They will not tell you that it codes for protein, but they will tell you that the coding region. So the coding region on the DNA does not just involve the area that codes for proteins. It also involves the area that codes for microRNAs. Okay? So that coding region of the DNA constitutes about 8 to 15% of the DNA. Are we together? The rest does not code for anything, and it is just junk. Okay? But if you want to look at the area, the regions of the DNA that codes just for proteins, is less than 2%. Okay? It's less than 2%. So about 75% of the DNA is junk. Okay? Maybe with time, as research goes on, they will discover another functions for that junk area of the DNA. Okay? So now, how does this stem lobe give rise to? How does this stem lobe that we saw up here, this stem lobe, how does it give rise to the other microRNAs? Oh no. Okay, so how does this stem lobe give rise to the other microRNA? It is cleaved, okay? It is cleaved by an enzyme known as dicer in the cytoplasm. So it will come, that dicer will come and cleave away this lobe. Are we together? It comes and it cleaves away that lobe. So we'll end up having these two strands. Okay? We'll end up having these two strands. And then this one above, the 5PN, constitutes the 5P microRNA that we have below. Okay? And this one below constitutes the 3P. Are we together? So they are somehow complementary. Are we together? Okay. So I'm saying that this stem lobe, okay, is the premature microRNA. How does it give rise to the other two microRNAs that we have below? That is the 3P and the 5P. What happens is that an enzyme known as DISA, okay, you have to look at the biosynthesis of microRNA. I think if you go to the course that I uploaded online, it is for molecular biology level 3. No, it's level 2, I think. Yes, it's level 2. Level 3, sorry, level 3. You will see the biosynthesis of this microRNA is better explained in that course. Okay, so that DISA will come and cleave this area here and remove this lobe. And when it cleave and remove this lobe, we'll have just these single strands that are paired up together. Okay, and I'm saying that they will unfold in a later stage in their biosynthesis. Okay, they will unfold, and when they unfold, this upper strand will give rise to the 5P microRNA, and this one below will give rise to the 3P microRNA. Are we together? Okay, so that will give rise to this 5P and this 3P. But we should notice that when we see that, it is not as if microRNA is double-stranded. Okay, it is just that it has formed a lobe and then unfold and then fold on itself and form that complementarity. Okay, voila. DICER, D-I-C-E-R. Before DICER acts, there is another enzyme that acts on it, on the pre-microRNA. The pre-microRNA, the enzyme that acts on it is DROSHA. DROSHA acts on it, cleaves it to give the premature microRNA. Okay? Yes. So those terms you have to go and look at it in order to understand. It is a whole process. I don't want to get totally into it, but I just want you to know that it is not only DICER that acts in this process. You have other enzymes such as, before DICER acts, there's another enzyme known as DROSHA that acts on it, okay? So you should look at the biosynthesis of microRNA. So our question is saying that we should download or copy the nucleotide sequence of this microRNA from MiBase Release 21 database. But now we have seen that the site is upgraded to MiBase Release 22 database. Okay, so the second question says, also copy the sequence of the other arm of this microRNA. So the other arm they are talking of is this, the 5P, the HSA Me29A5P. So you people, you copied that order, the sequence of that first microRNA, or this second one. You should click on get the sequence and copy it so that we follow our question as it is on the form. So when you click on this get the sequence, it will give you the sequence of this 3P.
Because we got that for 5P already.
Sequence of 5P? Yes. So if you want the sequence of 5P, you will come here and click on this get the sequence. Okay? I don't want to click so that it will be delaying my time. You can still copy this, but it's that it will give you in the lowercase letters. Okay? But since we want it in uppercase letters, you can click get the sequence. Are we together? Or you can copy this and then use your computer on Word to put it up to uppercase. Any way you choose. So we have the two sequences. Then what conclusions can you draw? Are they the same, different, or complementary? We already said they are complementary, not so. If we look at this, is what I was explaining now. That is how microRNAs are formed. The 5P and the 3P are usually complementary because they are coming from the same stem lobe. And that stem lobe is the same microRNA that folds on itself. Okay? And it folds on itself because of the formation of these complementary bonds. Are we together? Voila, so they are complementary. I just wanted you to know that it is complementary. That is why that question is coming in. Okay?
So are we comparing
5P with just the Mi-29A or 5P and 3P?
5P and 3P.
What? Follow the question as it is on our form. Okay? We started by saying that what? We start from the beginning. The human microRNA, that 3P, not so, that is HSA, Mi-29A 3P, has been reported to be upregulated or overexpressed in tuberculosis patients compared to healthy controls. That is our kick point. It is because this microRNA has been overexpressed in those that have tuberculosis compared to healthy individuals. Okay? So it might be that it is some sort of a biomarker, this microRNA. So that is why we selected this microRNA. Okay? Question 1 is telling us that download or copy the nucleotide sequence of this microRNA from MiBase Release 21 database. They have given the link there. Okay? By searching with the entry HSA Mi-29A. If you search this, it will still give you this information. Are we together? But I was saying that here there's a mistake. There's a typing mistake because he instead writes HAS. Okay? So we have downloaded the sequence for that our microRNA, that is 3P. Okay? Question 2 says that also copy the sequence of the other arm of this microRNA. That is, they have given you the other arm. The other arm now is this 5P. No longer 3P. But before now, we have already downloaded the sequence for 3P. Okay? The person who asked the question, are you there? Good. Then, compare the two. Compare the two, that is 3P and 5P. What conclusions can you draw? Are they the same, different, or complementary? And we have said that they are complementary, not so. Voila. So we move to the next question, 3. Also retrieve the sequence of the immature microRNA. That is, they see another error there. Okay, it should be HSA, not HAS. HSA, Mi-29A. Here you see at this, you see that Mi has a small r. The Mi there has a small r in question 3. Are we together? The Mi there does not have a capital R, it has a small r. Have we seen? And it comes back to what I was telling you, that it is the premature, that stem lobe is the premature. Okay? That stem lobe is the premature microRNA. So, to distinguish it from the mature microRNA, they usually write it with a small r. Are you confused? Because I see you searching. What are you looking for? Are you there? Or are you confused?